Okay. Okay, let's start our next talk. We have Dr. Jinmyo Chen from National University of Singapore with us, and she'll be talking about AI and spatial omics analysis. So give her a round of applause and let's start.
Hello, good morning, everyone. Thanks for coming to my talk, and thanks the organizer for inviting me. So today I would like to talk about AI-enhanced integration of spatial omics. So let's start with spatial transcriptomics that measures the gene expression together with the spatial locations of each cells or each spots. So how do we acquire the data? We usually cut the tissues into very thin slices and then place it onto the SD slice, wherein the captured spots contains the spatial barcodes that will be sequenced together with the MI molecules. So the data we obtained as an output will be a gene expression matrix that contains the genes in the columns and the spots or cells in the rows, as well as the spatial locations as the X and Y coordinates. So there are many methods allows us to profile the spatial transcriptomics. For example, we have the 10x Vision and Vision HD. Both are sequencing-based technologies, means that it is untargeted. It will capture all the MI molecules that are present in the tissue. And Vision provides a spatial resolution of 15 micron, and HD is higher, 2 micron. 10x, we also have another technology called Xenium, which is imaging-based. That gives us a much higher spatial resolution, allowing us to zoom into the cell to measure the location of different individual molecules, MI molecules within the cell. But this technology is a probe-based, imaging-based, so it can measure predefined gene panels of like a few hundreds or thousands or even like tens of thousands of genes. In addition to Xenium, we also have the COSMAX SMI or the Vision MERS scope to measure the MI expressions with imaging technology, probe-based, and this also gives us a subcellular resolution. With all these data, we require very precise cell segmentations to assign the molecules to the correct cells. Lastly, we have the StereoSeq technologies developed by BGI, which is a sequencing-based, untargeted, and also provides a subcellular resolution. So with all these data generated by spatial transcriptomic technologies, the real bottleneck or challenge lies in the data analysis. Different from the single-cell omics data analysis, we need to perform the analysis in a spatially aware manner, meaning that we need to incorporate the spatial information into the analysis. Just like the single-cell omics data, we will take a few key analysis steps. First step is to impute or denoise the data, because spatial transcriptomics also suffers from data sparsity due to dropout events. A lot of MRAs are expressed, but the technology is not sensitive enough to detect all the expressed MI molecules within the cell. So most of the time, we need to impute and denoise the data. After that, we reduce the dimensionalities to capture the major variations in a low-dimensional representations, to reduce the original a few hundred, a few thousand, or tens of thousands dimensions down to maybe 50 or 100 dimensions, where we can better group the cells into clusters. After clustering, we perform differential gene expression analysis to identify genes that were upregulated in a cluster versus others, and based on differential expression genes, we annotate the cell types or tissue substructures. Lastly, we infer the cell-to-cell interactions based on the ligand and receptor interactions and based on the spatial proximity. So spatial transcriptomics has been widely applied to all aspects of biology, for example, developmental biology. In this example, our collaborators in BGI use StereoSeq to profile the transcriptomes of mouse embryo at 8 different stages from E9.5 to E15.5. And to better analyze the data, we developed a method called STEM, which produces biologically meaningful low-dimensional representations of the data. Each low-dimensional, we call it a topics, is assigned with a gene module where the genes are ranked from high to low based on its contributions to the respective topics or dimensions. So based on the gene modules, we can assign the topics or assign the dimensions to either cell types or like tissue domains. That's how we give biological relevance to each of the topics. So we apply STEM to this data to review like about 30 different topics or dimensions. Each topic represents either cell types or like organs. And most importantly, we look into the gene modules to see whether they can capture the gene module differences across different organs and during the different development stage. So we look into the UMAP, we found that the gene modules mainly clustered by organs, indicating the gene modules actually captures the major differences that can separate the different organs. And we look at the PCA of the gene modules, we see within each organ, each color represents one organ, there's a linear trajectory from early time point to late time point that suggests the gene modules actually able to capture these subtle differences across different developmental stage within each organ. Another capability of STEM is to deconvolute spatially overlapping phenotype. In this case, fetal liver is the major organ of hematopoiesis. It's also a place where hepatocytes are matured and developed. So if we use a canonical clustering algorithms, we might assign all the spots or cells in the liver into one cluster. But here, STEM would deconvolute the spots in the liver regions into two topics, one for the hematopoiesis and the other one for the hepatocyte development. And the corresponding gene modules were able to map to the relevant biological process for each of the topics. For example, for the hematopoietic topics, we see the genes enriched for erythrocyte development or myelocyte development. For hepatocyte topics, we see some liver functions such as the metabolic, lipid metabolic and alcohol metabolic. And the next application widely has been applied for spatial transcriptomics is the cancer biology. In this data set, scientists from NanoString apply COSMAX to profile the human non-small cell lung cancer by clustering analysis to identify different cell types and by cell niche analysis to identify different tissue substructures in the cancer microenvironment. And again, we apply STEM to reduce the dimensionalities down to 15 topics. Each topic here represents either cell types or tissue substructures. For example, we have tumor edge, tumor interior, fibroblasts, as well as various subtypes of immune cells. And most interesting things we found in the STEM analysis is that it identified a cuff, cancer-associated fibroblasts, and deconvolute this cuff from the regular fibroblasts. And by spatial co-occurrence analysis, we found that cuff topics are mainly co-localized with the tumor edge, meaning that these two the cuff mainly are present in the tumor edge and stopping the immune cells from infiltrating into a tumor. And the further DG analysis and pathway enrichment also confirms the cuff identity of this topic. Another application for spatial transcriptomics is a neuroscience, because it seems to me that the spatial transcriptomics has been has best performed for the brain tissues. In this data generated by with a slice version 2 from the mouse hippocampus samples, we use STEM to identify different topics, and each topic represents the different regions in the brain, such as the CA1, 2, 3 regions, the DG regions, and LA regions. And subsequently, we collaborate with Lin and David Gates from Northwestern University, and they use spatial transcriptomics to identify new microglia mechanisms that drive amyloid beta clearance in humanized patients with Alzheimer's disease. So after we have learned the the power and application of spatial transcriptomics. Today, I would like to focus on the integration of spatial omics. So there are four major types of integration. The first type is horizontal integration, where the data comes from different tissue slices, but acquired with same technologies for the same omics modalities. Second type of integration is called vertical integration, and the data comes from the same tissue slides, but multiple omics modalities are acquired, such as transcriptomics or proteomics and epigenomics. And the last two types of integration involved are like diagonal integration or mosaic integration. So this integration integrates the data across sections and across modalities. Different slides have different compositions of the modality composition. I'll start with the horizontal integration of spatial transcriptomics. In this, the first scenario of data integration, we have the sample slides taken from consecutive sections from the same tissue samples, and these samples are profiled with the same technologies, text vision. And before the batch integration, we found the cells from the two sections are well separated, which indicates a substantial batch effect. Then we applied SVI, Harmony, and Stargate, as well as a GraphST to pull the cells from the two sections together, and then in the batch-corrected embeddings, we identify the clusters that are shared across these two sections. In the second data set, which comes from the human dorsolateral prefrontal cortex data, it contains 12 consecutive slides from the mouse cortex tissue samples. And again, before the correction, we can see the cells from different slides have some slide-specific clusters, and the different layers of the cortex cannot be well separated. But with the AI algorithms to remove the batch effects, we can evenly mix the cells from the 12 slides and well separate the 6 layers as well as the white matters. And the second scenario of data integration, I would call it is called like a horizontal stitching of multiple sections to reconstruct a bigger tissue samples. Because in a lot of times, our tissue samples are bigger than the ST capture slides. We need to cut the tissues into multiple sections and perform the spatial transcriptomics separately, and then we stitch the data, different sections, at the data analysis stage. So in this example, scientists cut the mouse brain into anterior and posterior brain and perform the 10x vision profiling separately. And later, we integrate these two sections at the data analysis steps. We apply GraphST, Stargate, and the SPA GCN to remove the batch effects across these junctions to identify the common structures as well as the unique structures for these two sections. And most importantly, we found that those are common structures. For example, the cortical layers are well aligned across the junctions, which indicate that the batch effects were effectively removed. And the third scenario of the data integration is very critical when we try to build a spatial tissue atlas, where we need to integrate the data of the same tissue types, but generated from different labs, different studies, and with different technologies. So here we look at the mouse olfactory bulb data generated with three different technologies. The cells was well separated by technologies before the batch correction. After batch correction, we are able to mix the cells to a certain extent, but we still see some technologies specific clusters because the batch effects due to these technology differences is quite substantial. And after the batch correction in the low dimension embeddings, we perform a clustering. We were able to identify the 7 laminal layers in the olfactory bulb. Most importantly, we align those 7 layers across different technologies. So this is very important for us to remove the technical variations when we are trying to build a spatial tissue atlas by integrating data from different labs and different studies, even different technologies. So, and then the second types of integration I would like to talk about is the data acquired from the same tissue slide, but have more than one omics. And such data comes from a technology called spatial multi-omics, which simultaneously measures more than one omics, such as transcriptomics, proteomics, chromatin accessibility, and histone modifications from the same tissue slides. We know that different omics layers have different and complementary information about the tissue's biology. So we hope to integrate these complementary information to have comprehensive characteristics of the tissue and to have a higher resolution of the tissue heterogeneity representation. So many technologies out there for us to profile spatial multi-omics. The first types of technologies simultaneously profiles the transcriptomics and the proteomics. Technologies include like sports and DBI-seq and spatial size-seq developed by Liu Yang in Rongfang's lab. Besides this technology developed in an academic lab, we also have many commercially available technologies such as the 10x vision, 6-axis spatial gene and protein expression, COSMAX SMI, stem-cell multiomics, and the still size-seq. All three technologies are commercially available and allows us to profile both the RNA and the protein from the same tissue slide. And besides RNA and protein, epigenomics is also very important. So Deng Yanxiang from Rongfang's lab has developed spatial cut and tag to measure the histone modifications in a spatially resolved manner. And the spatial ARTAC measures the chromatin accessibilities from a tissue. And later on, Zhang Di from Rongfang's lab developed spatial ARTAC RNA-seq and the spatial cut and tag RNA-seq to co-profile RNA with either the ARTAC-seq or the histone modification. And more recently, Yanxiang's lab developed this spatial DMT, which simultaneously measures DNA methylation and the spatial transcriptomics from the same tissue slide. I haven't looked at the data yet, but I think it'd be very interesting to look at how the DNA methylation can be correlated with the gene expression activities. And actually, we can go beyond more than two modalities. So, for example, we have this DBI-seq, ARP-seq, can measure RNA, protein, as well as the chromatin accessibility. And the DBI-seq CTRP-seq can measure RNA, protein, and the histone modification. And this paper has been accepted by a journal and is developed by Zhang Di from Rongfang's lab. And beyond that, Deng Yanxiang's lab has developed methods called spatial mask that measures 5 modalities from the same tissue slides, including the RNA, protein, chromatin accessibility, and the 2 additional histone modifications. It seems that we can go like more and more omics layers from the same tissue slides. So with all these additional layers, the data become multi-modality, and the complexity is so high. How do we integrate these different omics information in a spatially aware manner? So this is a real challenge, and that's why Yong Yahui, who was previously my postdoc, and he developed these methods called spatial glue. It is a graph attention neural networks. It adopts 2 layers of attention. At the first layer attention, it integrates the spatial information with the omics measurement. At the second level of attention, it integrates the representations from the different omics measurement. So by using 2 levels of attention, spatial glue integrates the different omics layers in a spatially aware manner. We first apply a spatial glue to these spatial epigenomics and the transcriptomic co-profiling data generated from mouse spring tissues using Zhang Di's technology. The data has RNA modality as well as the ARTAC modality. With the RNA alone, we can nicely segregate these CCG regions, which represented by the salmon color, but we couldn't delineate the cortical layers in the mouse brain. But when we look at the ARTAC modality alone, we can better separate the cortical layers, but we couldn't delineate the CCG regions as nicely as the RNA modality. So we use a number of algorithms, including SUREC, MOFA, MultiVI, and the step map and spatial glue to glue the informations across RNA and protein ATAC modalities to have a better delineation of the different brain structures, including the cortical layers, as well as the CCG regions and a few other brain substructures. And the second data set we apply spatial glue is obtained with the BGI's stereocyclic technology on the mouse thymus samples. The data contains the RNA and the protein modality. With RNA and protein modality alone, we can identify some structures, for example, the medulla or the cortex layers. But when we integrate these two modalities with AI methods, including spatial glue and Mephisto and a number of others, we can increase the resolution of the tissue structure. For example, we can identify the medular structures, the cortical medular junctions, and the different layers of the cortex. And each layer contains a different subset of immune cells, in particular the T cells. And the last data set we applied to is the 10x Vision 6-assist spatial gene and protein expression data. This data was generated by David Gates from Northwestern University using a human lymph node sample. And the link from David Gates' lab is very nice to offer us this manual annotation to serve as a ground truth for benchmarking a number of clustering algorithms. And the benchmarking shows that the methods were able to recapitulate the manual annotations. And lastly, I would like to talk about a mosaic integration, which is the last type of integration, and which is also the most complicated. So in a mosaic integration, different sample sections have different and complementary omics compositions. For example, section 1 has RNA and protein, section 2 has RNA and ATAC, section 3 only have ATAC, and section 4 have RNA and protein and metabolism. So this layout is very mosaic. So we like to develop an AI methods to achieve a consensus domain identifications across all the integrated sections. Meanwhile, we also wish to enhance the signal-to-noise ratios to improve the domain identifications by leveraging the signals from other sections or from other omics. And lastly, we wish to impute the missing modalities so that we can investigate the correlations of two omics that cannot be co-profiled together. So this is the method we developed called a SPA mosaic. I'm not going to the details of the techniques. And in essence, it adopts a contrastive learning and the graph neural networks to build a modality agnostic and a batch corrected latent space. So with this latent space, we can perform the downstream clustering, imputation, and interpretation. So with this modality agnostic and batch corrected latent space, we can borrow the information from other sections or from other omics to enhance the signal to noise ratios for the sections under study. We first applied this SPA mosaic to the data set generated by MISA-6 that was generated by Peng Guangdun's lab. So MISA-6 can co-profile RNA and chromatin accessibility from the same tissue slide. And we took the data from 3 developmental stages, E13.5, 15.5, and 18.5, and artificially remove the ATAC modalities from 13.5 and RNA modality from the 18.5 to mimic a mosaic configuration. And when we look at the data before integrations, the samples from different stages have very different sample size, shape, and even orientations. And all these heterogeneities or difference in the samples pose a significant challenge in the data integration. But we managed to integrate the data across omics and the samples to have a consensus domain identification. These methods include a CLUE, COBOT, MIDAS, MultiVI, SEMOMAT, StepMap, and the spatial SPA-MOSAIC. So this method, some of the methods were developed for single-cell mosaic integration, wherein spatial information is not included. So we can see in the results, right, the clustering was slightly noisy because they didn't consider the spatial information. But for SPA-MOSAIC, we integrated data in a spatially informed manner, so it was able to improve the spatial smoothness significantly. And next, we want to investigate whether the clusters is meaningful. So we zoom into one of the clusters that represents the inventricular zones of dorsal pallium. We plot the RNA expressions of the marker genes as well as the gene activity scores inferred from the attack modality. So, and then by looking at this expression as well as the gene activity scores, we confirm the identity of these clusters, and then we cross-check with the human Allen Brain Atlas to further confirm that these clusters is actually the T-Pal region. And next, we apply this to another data set acquired from postnatal mouse brain sections with very diverse epigenomic layers. The data was generated by Rongfang's lab, and it contains 4 sections. Each section has the RNA modality together with 1 epigenomic layers. So we have 3 histone modifications plus 1 attack modality. So each of the sections have different epigenomic layers. So we wonder whether we can perform the mosaic integrations to have a better separation of the mouse cortex layers, and the result shows yes. Although some methods it's not very good at, it has relatively noisier representations, but we are very confident that by mosaic integration, we can improve these resolutions when we dissect the tissue heterogeneity. So as I mentioned before, we would like to impute the missing modalities. So what we did here, for the section 4, which contains only the RNA and attack modality, so we use AM algorithms to impute the 2 histone modification modalities. Then we examine the correlations between the RNA expression and the gene activity score inferred from the imputed modalities compared to that correlations from the measured modality. What we found is that in the imputed modality, we have more genes shows positive correlations, and those genes that shows correlations in the measured omics modalities were also found in the imputed modality, but with imputation, we managed to identify more correlated genes, and then these unique genes will also have biological meanings. So, and then we apply this to a more complicated integration, which contains 6 samples from different stage and comes from 2 different technologies, SteelSeq, MesaSeq, and the samples highly variable in terms of size, shape, orientations, and developmental stage. And the technology have different spatial resolutions, and number of cells range from tens of thousands to thousands. So data is very, very heterogeneous. So we still managed to integrate the data and correct the batch effects to delineate the different regions in the mouse brain, mouse embryo. And the last is the comprehensive integration that contains 7 sections, have 5 different modalities. And the technology includes 4 different spatial omics techniques. So we also test the different methods, and then found that it was actually able to remove those batch effects and then identify the different omics in different sections. And then lastly, I would like to mention the scalability. So it was able to integrate the data more than 100 sections within 30 minutes, and each section allows to have more than 800,000 spots per cell. And lastly, I would like to mention about this the atlas concept, because we all know that scientists are building this single-cell atlas to better understand cellular complexity. And by integrating the data across studies, across technologies, we can uncover new cell types or rare cell types, and also pinpoint to the disease cells for precision medicine. However, most of the existing single-cell atlas lack tissue spatial context in the tissue, because when we perform single-cell, we need to dissociate tissues into single-cell suspension, and during that process, we lost the spatial context of the cell. So we would love to construct a spatial tissue atlas through data integrations from the same tissue types, but different samples, and different samples may have different sample characteristics, for example, size, shape, orientations, and stage. And then the data may come from different labs and different studies using different technologies. And these technologies has highly variable spatial resolution, number of spots or cells, or like they have also top of different gene panels. And then the data also may come from different modalities, so including the transcriptomics, epigenomics, proteomics. So this data is highly, highly diverse and heterogeneous. So this poses a very significant technical challenge when we try to integrate data. First thing is the batch effect caused by the non-biological variations. Another challenge is the scalability. How are we able to integrate like thousands of samples, or even like tens of thousands of samples to construct this tissue atlas, like what we did for the single-cell atlas, which integrates like maybe thousands of samples to construct, to have like tens of million cells in the atlas. So this scalability is another key challenge if we want to build this spatial tissue atlas. Another question I always ask myself is that when we build this spatial tissue atlas, does it mean that we only need to detect the cell types or tissue substructures? What else we can do more? So for example, with the spatial context, we can look at the cell-to-cell co-localizations. And with that, we can identify cell niche or cell-to-cell communications. But how do we integrate such informations from the spatial data when we are building this spatial tissue atlas? I think these are all open questions, and then this requires the collaborations between bench scientists as well as bioinformaticians and AI experts to come up with like very powerful and very scalable methods to integrate currently all the available spatial omics data to come up with the spatial tissue atlas. So lastly, I would like to thank my lab members and collaborators, and thank you for listening. Thank you.
We have time for a few questions.
Hi, thanks for your nice talk. And my question is that when we process different modalities, they usually have different resolution, like there are pixels or segmented single cell or some spots. How can we align this basic unit? And they often have different like gene numbers or protein numbers. They may vary a lot. And will that affect the performance of this integration? Thank you.
Yeah, thank you for your question. That's a very good question. So I don't think the existing integration is perfect or optimal, including those developed by my lab. So regarding your questions about like different resolutions, right? Like spots versus cells, or like 50 micron versus 2 micron. So my idea is just my idea. So we want to do this alignment-free integration. So we don't force the spots to match the cells, or we don't force the 50 microns, or we don't sum up the 2 micron spots to represent 50 microns. I don't think that is just my idea. So I don't want to do this like alignment-based integration. What we want to do is do like alignment-free integration. So we keep the original resolutions. If it's spots, leave it as a spot. If it's cell, we leave it as a cell. And then in the integration, right, for example, in UMAP, each dot still represents the original spots, original cells. So we don't force them to align and don't force them to become a same spots or same cell spatial multi-omics data. That's what I think a better implementation.
Thank you very much.
Yes.
Hi.
Yeah.
Very cool work. So my question actually is related to both spatial glue and the new SPA mosaic. Right, so both of these approaches have attention mechanisms, right, in their background. And then what you showed us today is that you can nicely identify the different anatomical regions or niches, right, based on integration of multiple mixed data. So my question here is, if I want to look deeper into one of the identified niches, right, based on the representation, can I extract the underlying relationships between the different modalities from these models, and to what extent?
Yeah. So that's also another very good question. So we look at spatial multi-omics with one key goal is to look at the cross-omics correlations and to see how the different layers communicate with each other. But I have to admit that even in spatial glue, we haven't done so much towards that yet. So that's why I say that beyond cell types or beyond domains, how deep can we go further to look at, okay, so for example, we identify these, the regions, 4 CCG regions, and during developmental, we can see these regions like evolve from one stage to another, to the next stage. But can we like look into these like multi-omics to look at this omics link? I think there's still a lot more to be done. And another point I would like to mention is that in the mosaic configurations, because due to the technology limitations, certain omics cannot be co-profiled simultaneously, cannot co-profile. So we have to do some kind of like diagonal integration. There's no shared omics modalities. But if we have some multi-omics data from the same tissue slabs, tissue type from another study, we can use that as a bridge to integrate these diagonal omics and then to impute the missing modalities so that we can have the 2 omics measurement that is very, very difficult to co-profile. And then with that, right, that will give us a new opportunity, a new ways to look at the omics to omics relationship. That's what I think the AI can do something in that aspect. Thank you for your question.
Hi, Dr. It's a very nice talk. Yeah, exciting. I'm Shaokun Shu from Peking University. I work on this. I'm a biologist. I work on the multi-omics study for the drug response and drug mechanisms. So my question is that, looks from here you integrate the multi-omics data. Looks like you didn't integrate the staining pictures.
The H&E image.
Yeah, HE or immunofluorescence staining pictures or other image pictures. It's hard? Or?
Yeah, it's hard for my lab.
Oh.
I apologize because for spatial omics, right, there's another 2 additional layers, which is an image, for example, the H&E image. But we have many scientists here in the audience, including Li Mingyao, who works on digital pathologies to integrate the H&E image to better analyze the spatial omics data. So I think that's a very, very good question. Just because of the limitation of my lab, so we tried, but we didn't manage to. But it is a very important layer to integrate into the omics. Thank you.
Sorry, it's a naive question, just biology. So, and further question is whether you, after integration, can you build up a 3D model?
Yes. So that okay. So So with spatial, we can only look at 1 2D sections,
2D
right?
section.
So it's very important to obtain many slides from the same tissue samples and then computationally integrate them to construct a 3D spatial omics. So a lot of methods have been developed and a lot of papers have been published. And then, for example, Jin Yang from Sydney University, she has these review articles about this 3D reconstruction of spatial omics. So I think there are many methods out there, and then the essence of those methods utilize a method called optimal transport. Optimal transport. It also involves this optimal alignment of dots or spots across sections in the Z stack.
Yeah.
So when they manage to align the spots across the sections in the Z in the third dimensions, they can reconstruct these 3Ds. And then I would like to also highlight that besides this computational reconstruction of the 3D omics, right, there is a real-time, real 3D spatial omics. Means that they use some chemicals to clear the tissue, to make the tissue transparent.
I know that, yeah.
I know that.
Or something, yeah.
Yeah, so but...
My point is that for the first question, whether you just do the 2D integration first and build up the 3D, or you just directly build up the 3D multi-omics.
3D for now cannot do high perplexity. So that's based on my knowledge. So we only can profile a handful of protein or gene expressions. So, but at this stage, maybe we can think about how do we integrate the high perplexity 2D spatial omics with the real 3D low perplexity, and use the AI to have this like cross technology integration and imputation to have a real 3D spatial omics. I think it is possible. Yeah.
Okay. Thank you.
Thank you.
Okay. Let's thank Jinmiao again for the wonderful talk, and we can continue more discussions in the lunch break. Let's welcome our next speaker, Xiaomin Ying. She's a professor at Beijing Institute of Basic Medical Sciences, and she'll be talking about how to build interpretable and dynamic models from genomic data.
Okay, good morning, everyone. Thank Dr. Chen for inviting me, and thanks Xing for introducing me. It's my great honor to be here to share my work. My name is Xiaomin Ying, and today I will be talking about insight over scale, building interpretable and dynamic models of biological systems from single-cell sequencing data. This is the era of large models, such as ChatGPT, Gemini, and DeepSeek, which are fundamentally transforming our life and work. Large models are the result of what I call brute force aesthetic, a convergence of massive data, advanced infrastructure, and huge computing power. Just look at the scale of data. ChatGPT-3 was trained on around 500 billion tokens. Newer models like DeepSeek V2 use over 8 trillion tokens. Llama 3 use 15 trillion. Then, the open question is, are we ready to replicate the success of large models in biology? We have over 130 petabytes for genomes, 15 peptides for transcriptomes, and 1.5 petabytes for proteomes, and sequencing data from over 120 million single cells. Are the data volume, the existing infrastructure, and the computing power sufficient for the same brute force approaches? As is known, biological systems are far more complex than language and images. DNA, RNA, and protein all undergo many kinds of modifications. RNA also undergoes alternatively splicing and editing. Crucially, all these molecules exist and function within complex 3D structures, and the entire system exhibits intricate spatial-temporal dynamics. One bottleneck for building large multi-omics models in biology is the scarcity of massive paired multi-omics data. In practice, available datasets are typically unpaired and mosaic-like, suffering from pervasive missing omes and features. Our first study is integrating large-scale mosaic data with MIDAS so as to generate massive paired multi-omics data. The core insight behind MIDAS is that the counts detected by single-cell sequencing are actually a mixture of the cell's true biological state and the various technical noises introduced during the experiment. Based on this insight, we built a deep generative model. We assume that for each cell, the observed data and its batch IDs are generated from two independent, modality-agnostic, low-dimensional latent variables, biological state C and technical noise U. The model could be trained with datasets. MIDAS uses the VAE to implement model learning and latent variable inference. Self-supervised learning is used to align different modalities on latent space through joint posterior regularization, and information theoretic approaches help disentangle the latent variables. We trained MIDAS by summing the total losses of generative modeling loss, modality alignment loss, and information bottleneck loss. Once trained, MIDAS offers several key functionalities: simultaneously dimensionality reduction, imputation, and batch correction of mosaic data. We first evaluated MIDAS on rectangular integration tasks, where all batches contain all three modalities. We construct two trimodal human peripheral blood mononuclear cell data. These UMAP plots show the latent variables inferred by MIDAS on the dogma-4 dataset. The biological state C effectively separates known cell types while mixing cells from different batches. Conversely, the technical noise U clearly separates the batches, but shows no cell type specific structure. This visually confirms the success disentanglement. Correlative comparisons of MIDAS and non-rectangular integration strategies show that MIDAS outperforms other methods in batch correction and cell clustering. Quantitatively, using the standard SCIB benchmark, MIDAS achieves the highest scores across batch correction, biological conservation, and overall score, significantly outperforming competitors. Next, we tackle the more challenging trimodal mosaic integration task. We constructed 14 different mosaic datasets by systematically removing modality batch combinations from the rectangular dogma and T-dog datasets. Even when trained on mosaic data, MIDAS effectively learned aligned biologic states across modalities and batches, and successfully isolates batch-specific technical noise. The joint embedding space integrates information robustly. Quantitative comparisons of MIDAS on rectangular data and mosaic data show that MIDAS performance on various mosaic tasks is remarkably consistent and comparable to its performance on the 4 rectangular data. This highlights its robustness to missing data. We compared MIDAS with 4 other methods capable of mosaic integration strategies. Qualitatively, on the complex dogma diagonal +4 task, MIDAS maintains clear separation of cell types while mixing batches effectively. The other methods struggle with either batch effects or preserving biological structure. Quantitatively, MIDAS consistently achieves the highest overall scores across all tested mosaic scenarios. MIDAS enables atlas-level integration. We collected a large mosaic dataset of human PBMC comprising 9 trimodal batches and 18 bimodal batches from 10 different sequencing platforms, totaling over 185 cells. The results show that MIDAS can effectively incorporate more cell types and omics, aligning cells from diverse technical origins into a coherent biological space. This integrated atlas allows for more comprehensive cell typing and facilitates the discovery of real cell populations. We further applied MIDAS to construct a trimodal atlas of human bone marrow mononuclear cells with continuous cell state changes. The MOSAIC dataset is composed of 3 batches of bimodal data covering over 7,100 cells. The integrated UMAP reveals distinct lymphoid and myeloid counterparts. Pseudo-analysis of RNA missing data based on latent variables and imputed data from MIDAS reveals the trajectory starting from hepatotic stem cells and branching to branches. It's worth noting that with MIDAS, we are able to computationally break through the modality limitations of current sequencing technologies. By integrating data via different modalities, we can create a more complete picture, which is especially valuable as we move towards building large multi-omics models. Indeed, in a very recent comprehensive benchmarking study published in Nature Methods from Professor Liu Qi, MIDAS was ranked #1 for bimodal mosaic integration and is highly recommended for these tasks as well as cross-modality imputation. MIDAS effectively addresses the challenge of integrating mosaic datasets, but biological data is rapidly growing. This leads to our next question. How can we efficiently and continually integrate single-cell multimodal mosaic data to build massive paired multimodal datasets? Data is being generated continuously and rapidly, emphasizing different biological issues, for example, different disease stages, different development stages. This brings us to our second story, Miracle, a framework for the continual integration of single-cell multimodal data. Most existing integration methods operate offline. This means whenever new data arrives, you have to reintegrate the entire collection of old and new data from scratch. As datasets grow, this becomes computationally prohibitive and hinders timely updates. Online integration methods are emerging to tackle this. The methods are categorized into 2 types, that is, generalization-based methods and fine-tune-based methods. Generalizing-based methods quickly mapping new data onto pre-built reference atlas. They are fast but struggle to incorporate novel knowledge from new data. Fine-tune-based methods are moderately fast and adaptable, but they are struggling to knowledge forgetting. Furthermore, no online integration methods has been reported for mosaic data integration. To overcome these limitations, we developed Miracle, multimodal integration with continual learning. It's an online learning framework designed specifically for single-cell multimodal mosaic data. Miracle incorporates 2 key continual learning strategies: dynamic architecture adaptation and data rehearsal. The effectiveness of rehearsal depends critically on the sampling strategies used to maintain the memory within a limited capacity. Simple random sampling can distort the data distribution, especially for rare cell types. Therefore, we developed distribution-preserving reservoir sampling. It combines stratified sampling to preserve proportions across batches with a novel intra-batch sampling method called ball tree sampling. We benchmarked the BTS against simple random sampling. Sketch and SC sampler, BTS consistently achieves the lowest maximum mean discrepancy, indicating the best preservation of the original data distribution, particularly at lower sampling ratios. It's also significantly faster than Sketch and SC sampler, comparable in speed to SRS. We tested the Miracle on a large human data set. We found that Miracle integration performance improves with memory capacity, saturation around 20K memories, just 1/26 of the total memory size. Just use 20K memory, BTS Miracle achieves the performance comparable to the offline gold standard and significantly outperform Miracle Transfer, Miracle SRS, offline PCA, and the pre-trained model GeneFormer. Qualitatively, the UMaps shows Miracle BTS 20K effectively removes batch effects and preserves cell types similar to Miracle offline. Importantly, the cumulative runtime of Miracle BTS 20K grows linearly with batches, whereas the offline approach's runtime grows quadratically, demonstrating Miracle's superior time efficiency for continual updates. We then evaluated Miracle's ability to handle sequentially arriving batches with variable cell type. The UMAP visualizations over 8 time points show Miracle progressively integrating new batches, while maintaining the structure learned from previous batches, effectively adapting to changing cell compositions without forgetting. Comparison confirms that rehearsal is essential. Miracle performs similarly to offline, while Miracle Transfer fails to mix batches properly. Against 11 other methods adapted for online settings, Miracle achieves the highest SCIB score, demonstrating its state-of-the-art performance for online bimodal integration. A key advantage of Miracle is its ability to handle mosaic data continually. We tested this using the datasets with varying modality combinations across 8 batches. The UMaps over time show Miracle successfully integrating these mosaic batches, maintaining consistent biological representations. In contrast, adapting other mosaic methods like multigrade using standard transfer learning leads to significant performance degradation and failure to integrate properly as more batches arrive. Quantitative comparison confirms Miracle achieves the highest overall performance in online mosaic integration, substantially outperforming competitors and performing comparably to its offline counterpart. Miracle enables the continual construction and the refinement of large-scale atlases, even across different tissues. Starting with our comprehensive PBMC atlas, we sequentially integrated data from torso, bone marrow, and the spleen. Miracle effectively incorporates these diverse data sets, aligning shared cell types while correctly positioning tissue-specific populations like germinal center B cells in the tonsil or progenitor cells in the bone marrow as distinct clusters. In conclusion, Miracle provides a robust framework for single-cell data integration. It enables continual mosaic integration, addressing both data adaptation and knowledge retention through continual learning strategies like rehearsal and dynamic architecture. It achieves accurate online integration with significantly reduced computational requirements compared to offline methods, facilitating dynamic atlas updates across tissues and modalities. Its flexible model sharing and updating capabilities promote collaboration and democratize large-scale single-cell analysis, making atlas building accessible to more labs. Looking forward, building large multi-omics models in biology requires careful consideration. Modeling with biological insights, rather than relying on data scale alone, can compensate for data scarcity and may represent a more elegant approach. Generating and integrating more high-quality paired multi-omics data remains a vital step for building large-scale multi-omics models. Finally, reducing dependency on massive computational resources is key to enabling broader participation and broad research democratization. Okay, I'd like to acknowledge my key collaborators on these projects, and also I have very good collaborators, some PIs. I also thank our funding agencies. Okay, thank you for your attention. I'm happy to answer any questions.
Thank you for your talk. Don't have any questions. I'll quickly ask something. What do you think? How do you think the dynamics of these models, like how do we incorporate in the future the dynamics and how these processes, how they track over time? How do you foresee we incorporate those models?
I think it's very important issue, especially we want to build large multi-omics models in biology. As we know that we now construct a very large atlas with several billion cells. And when new data come, we will integrate those data into the old atlas that we have constructed. Then if we reintegrate from scratch, it will cost a very heavy computational resource and time. So our solution is to use using continual learning strategies to flexibly integrate those new data and facilitating the new knowledge from new data. I think it's a very important direction in this field. Although there are very few papers is seen recently.
Yeah, I think it would be important to, like how you said about continuous learning, like I think strategies to incorporate new data as it comes in with older established knowledge would be important.
Yeah.
Thank you. Okay, so let's thank Dr. Yang again. And let's welcome our last speaker, Dr. Kai Ye from Xi'an Zhaoting University. He'll be talking about genome sequence analysis framework for seq-to-image transformation.
All right, and thank you very much for invitation. And I changed it to the shorter slides. So it's a sequence to image, concepts, and applications. Okay, let me first get my... All right, so let me introduce the concept of the sequence to image. And as we know that reductionism is pretty important. So in physics, chemistry, and biology, so a complex system will be that we like to break them into a smaller, simpler pieces and try to understand the fundamental components and also maybe a simpler rules. And we actually also use it for the biology as well. So when we want to analyze a complex biological questions, for example, complex biosequences, so we tend to use the reductionism, build a simplified model. And however, because of the number of parameters is not sufficient, so often the case, we have to patch it. So with all kinds of the additional filters, increase the number of parameters. And let me give you one example here, the famous GATK. So GATK is a dominating variant calling method in the field, in genome sequencing field. It applied very elegant statistical model and to compare the probability that one base at a certain location to the background sequence error. And it's so simple, so little parameters. However, when it's applied to the real data, there's lots and lots of false positives. So at the end, what the GATK thing does is that they introduce 11, I think there are many additional filters as well. So they officially announced 11 filters, and they call it GATK best practice. So it's kind of, you have the higher simplified model, then you have the patches. So this is the way that reduction is done. So when we have this one in mind, and can we go away from this? Can we find a different way to do the size, not using the simplified model? So here what we are proposing is that instead of throwing away things, we add knowledge. So we add knowledge to the data. And we propose a sequence to image. We can use this kind of the data representation to code both data and knowledge. And then we can use the AI to try to extract new knowledge. So this is the way we can do better things. And also we can see that once we transfer the one-dimensional biological sequences to 2D images, it has extra space. We can code a lot more knowledge. And then when it's in the sequence, from the sequence space to the image, we can directly apply all kinds of neural networks and then make a prediction. So that's what I said is that image representation, we can code data knowledge. Then we can use the image operator to do all kinds of predictions. By doing that, we actually has a lot of benefits for the sequence genomic field. First one is that from the model-driven technology to the data-driven approach. And secondly, that we can use lots of the image processing technologies, which is not really available for the sequencing field. All right, so here's the concept of the sequence to image, and let me introduce step by step about some methods and also one applications. So the first thing is that we use CNN to clustering local features. So for this, in this application, so I will use the structuration. So basically, we convert the problem that computing the structuration from the sequence data to the image space, and we classify alignment anomaly for this one. So on the left side, it's a simple structurations with all kinds of the deletion, insertion, inversions, and all kinds of duplications. On the right side, it's so-called complex structurations. And by definition, that we know that we cannot explain it using the left side with just a simple sequence alteration, just a combination of multiple alterations. So when we want to detect such structurations or complex structurations from the sequence data, and there's several challenges. First one is that current methods, you first need a model and say what is a deletion looks like, what is insertion, what is duplication looks like. Then write a piece of code trying to detect such a mutational signature. However, for the complex event on the right side, and there's so many different kinds of complex events, and some mutations may show up in one of the rare cancer patients, but we haven't really detected yet. So that's the reason that we cannot use the model-based approach to find those things that we haven't detected. This is basically a chicken-egg problem. And secondly, that structuration often landed in the very complicated genomic regions. So sometimes those regions also can have a secondary structurations. So then we are going to see that the background repetitive signature and the mutational signature will mix together. It's really difficult to find out what's going on from the reference sequence to the alternative sequence. So there's no way we can find that. And so that's why we propose the so-called association approach, so convert everything from the sequence space to the image. So basically, when we compare two sequences, we can generate a kind of dot plot, but at the same time, we also introduce different colors. So we use color to indicate whether the segment is inverted or direct repeats, and whether it's extra copy or not. So by doing that, we actually code knowledge and together with the sequence alignment. And secondly, that remember, once we have the reference-to-reference alignment, this plot only contains the background repetitive signature. On the top image, and this is mutational signature plus the background repetitiveness. So we basically subtract these two images, then we can get a very clean sequence image similarity plot, only contain the information about the mutant. And then once we have this image, we can just straightforward using all kinds of CV technology, computer vision technology, to find the object and to do all kinds of classification. So this is the framework that there's an encoder, there's a neural network, and also one is the graphical representation of the mutant. And we can have the simple structurations as a training set. We convert the mapping result of the simple structuration into image, then train the neural network. And once the new sample comes along and a new variant comes in, then we can use this kind of alignment and convert it to this image again, and then use the trained neural network to do all kinds of predictions. So we only need simple structuration for training, but we are able to compute very complicated structurations that haven't been modeled for that. So here's the one example that we can finally find out is really beautiful. That this is the one very important in the neural development, CNTM5 genes. And with our approach, we are able to really find out there's a new complex structurations show up in the population, and actually very frequently among many major populations. But the reference allele is very simple, but only is private events. So this indicates that when we want to study a function of the gene, we may be very careful that what is the correct version, whether it's a private events, a private version in some individuals, or maybe we should choose the frequent ones. And so once we have the single sample calling, and next thing is that for the clinical applications, basically we have to compute between samples. So on the left side is what we already done. So we compare one sample against the reference. So we can write it as a difference between the reference and sample. On the right side, for the somatic events, for the de novo events, we have to compare different samples. For example, we have to compare the child with one of the parents. We have to compare the tumor tissue with the normal tissue. So this is two levels of comparison. So we first do a diff with the reference and do a second layer of diff with each other. So basically we know that the differences between two sequences is image. So we can basically substitute these two as the two image, and this has become the calculation of can we compute the differences between two images. And this is the game that we play very often, that imagine on the left side, it's a normal tissue you generated, and from the same patient. On the right side, it's the tumor tissue and tumor sequencing. And to detect the somatic events, basically we need to scan these two images and try to find the differences. And here we can see that there's 4 somatic segments, the ear, the tears, and legs, and so on. So this is the philosophy we actually applied in our algorithm part. So instead of we compare 2 images, we try to code the multiple sample information into one image, because when the previous representation of 2 sequences is quite empty, this plot. So we're able to code multiple samples into one image. For example, we can code the case genome information to the left corner, and the control genome information to the upper right corner. By doing that, we are able to code multiple samples and the alignment information into just one image. And we just need to compare different segments of the image. And because it's a real image, then we can have different channels, so we can have different colors. And that's the place that we can add a lot of information from the raw alignment. For example, we can code the coverage. We can code using different colors to code the alignment directions, whether it's a forward strand, reverse strand, or duplicated. Yeah. By doing that, we actually come up with such a presentation that this is the input, that we compress the alignment information from 2 samples into one image. On the right side, this is what we want to get. So we want to compute what's the case genome look like, what's the structuration looks like. On the upper one is the control genome. So we just need to find an AI program, AI model to make the gap. We test a few and found out this 1990s actually works okay. So this is the entire pipeline. And we actually did a lot of performance evaluation. So We can have the genotyping and de novo evaluation, also somatic evaluation. So most of the cases, I think that performs pretty well. And this work has been published on Nature Biotechnology this year. And so for more technical details, you can take a look. All right, so previously I mentioned we can use the CNN to clustering local features and genomic local features. And secondly, I want to demonstrate that we can also use transformers and to model distal interactions. And for that one, I come back to the really, really old genomic question, so about gene annotation. And as we know that, given a DNA sequence, and the question is where and how DNA sequence code functional elements. So basically, we want to learn the language of the DNA. And the current, there's actually actively developed for decades, and the current pipeline is actually from sample collection, sequencing, assembly, and it will take less than 1 month because sequence technology moving forward so quickly. However, for the gene annotation, it takes them more than 6 months. It takes running forever on the pipeline. And imagine that what's going on at the Darwin Tree of Life and Earth Biogenome Project. They're going to generate 70,000 species, whole genome sequences, and also 1.8 million species. So this is something like, I think is with current annotation pipeline, this is mission impossible. So that was the reason that we took up the task and tried to think about it by ourselves. So the first things that we have to tackle is that to model DNA, to understand the gene coding, we really have to model the long distances and also their distal interactions. And when we visit what is currently have in the field is that using the hidden Markov model. And this one actually only model the base adjacent bases, the base before that and base after this. But it doesn't really able to model very long distance interactions. So by doing that, we actually was using the high-C inspired modeling. As we can see that in the high-C representation, the DNA sequences and the interaction can be really easily modeled using the triangle-like density plot. And so we actually learned from this. And here what we did here, so we have a 32 base pair bin, and then we use a CNN-based local pattern modeling to extract information. And then we can use a transformer-based architecture to model the remote distance interaction between different beings. And okay, so then we are able to model the distal interaction. So the second challenge is that genes can have a different way to evolve. And the most common way is a vertical inheritance. And the gene can inherit it from the common ancestors and spread out from for different species. But there are also other ways for example, gene conversions, species can be hybridized, and there's all kinds of the horizontal-like and transfers. So that's why we have to handle both vertical transference and also the horizontal. So it's quite a complicated computational task that we have to consider lots of the species at the same time. So to tackle this question, the problem, so our solution is that we use a mixture of experts. And the idea is that since the tree, the gene, the species, they look like a tree, so the best way to capture the information from the tree is to use a tree. So we use the mixture expert. We basically generate a set of small models. Each individual model will capture a small lineage. And then we have an evolution relationship calculation. So really to assign different weight when we have new sequences come along. So by doing that, we are able to say, when you have new sequences, we can automatically say that for those models, how much weight we should assign automatically. Okay, all right, so this is a novel gene annotation framework. We have the contact extension, so we have the DNA sequences and also their franking sequence as well. And then we still use the neural network and to make the model, and then we can have the gene decoding part. Okay, to cut story short, let me show you the performance. And very interesting that, in the field it's actually heavily developed. So many pipelines and using the evidence base, so need the RNA-seq, protein, and also extensively require repeat masking. And there are also an additional method, for example, our ANOVA method. It doesn't need all this RNA-seq. It doesn't need, it's evidence-free. We don't need any RNA-seq. We don't need protein. We don't even do repeat mask. And we directly take the DNA sequence. Then we can output the gene structure. And our performance is even better than those need those evidence. And also because it's a lightweight model, and so we carefully choose the small model instead of running, we need a very large language model, so we choose a small model. By doing that, because we need to tackle the speed, and we cannot let the Sanger Institute to have a very expensive machine to run a large language model just to annotate one genome. So and we actually implement our system in a way that it can run on the laptop. And for example, we can annotate one Rhabdopsis genome just 3.5 minutes. And we can annotate one human genome just 1.5 hours. So they're actually game-changing in the field, and to basically it's 100 times faster than the current pipeline, and actually made it possible for the Darwin Tree of Life and also BioEarth program to move forward. And surprisingly that when we have automated annotation, it's very interesting that we actually able to correct those errors from the gold standard, because all these annotation were from the creators. They read literature based on and also gather some RNA-seq data and do the manual annotation. And in our automated annotation, we found out that actually, and for example, ensemble annotation that can contain lots of errors, and we made automatic correction. This is just one example that ensemble predicted as one gene, and we predicted as 2 separate genes. And all the RNA-seq data actually support our claim. All right, so I already mentioned about the sequence-to-image approach, the ideas, and also 2 computational methods. And last, I will present a very exciting application of the sequence-to-image. We use the sequence-to-image approach to analyze the evolution of the genome architecture. And so, okay, so you know that it's fundamentally the unicellular organism, and then this transition to the multicellular organism is one of the major evolutionary steps. And to do that, and there are actually many lineages, many successful stories. But there are 2 stories and 2 successful instances that are very prominent. The one is that from the unicellular organism to the animal kingdom, and the second one is that to the plant kingdom. And those 2 are evolving independently because they can different organelles. And so here's the question that as those unicellular organisms become multicellular, and how did their genome organize in 3D and to facilitate complex function? And secondly, since the plant and animal, they evolve independently, do they actually find exactly the same rule, or they find something different? So this is the question that we want to ask. All right, so we apply the sequence-to-image. So we obtained the data from the Darwin Tree of Life project, and they generate more than 2,000 species genome already, but at that time, we only have the 1,000 species, and we got the high-C data from it. And high-C data, by definition, is already in the image space. And then we apply the image operator to do all kinds of normalization, classification, quantification. All right, let me first introduce the data set. So we have all kinds of species from the bacteria, fungi, plants, animal, and in total more than 1,000 species. And with 100x high-C data is sufficient for genome scaffolding, but it's not sufficient to look at the TAD, to look at loop. And so that's why we have to do something else here. Okay, so the data normalization. So when we want to apply, for example, classification, when we look at the raw high-C data, it's really annoying that the diagnosed line that is dominating, but doesn't really tell us a lot of things. Simply because when you have 2 plots, 2 genomic locations bound by DNA filament, and a shorter distance will certainly give you a higher probability to bounce into each other than those long distance. So that's why I apply a filter to remove such a proximity bias. So we remove the distance bias. Then we have we call it normalized high-C data. But doing such an operation, you can we remove the short distance interaction and enhance the long distance interaction, but largely we made the biological signal remains the same. So then we go straight forward for the classification. So what we found that by doing all kinds of the more than 1,000 species classification, we found out there are two major folding categories. One is so-called the global folding. And when you look at it, it looks like the chromosomes fold by themselves, and the centromere goes one side, telomere goes the other side. This is actually well known in the field, in yeast, in bacteria, and so on. And also we found out there's another very widely adapted so-called checkerboard, vertical and horizontal lines. So this is actually correlated to the compartments. And so once we are able to do our classification, also quantification, so we can really look at the entire evolutionary tree to see how these two models, the intensity of these two folding genome architectures, how they evolve and along the tree. So what we can see here is that the global folding, it doesn't matter whether it's intercellular, interchromosomal, or intracellular chromosomal in global folding. It doesn't go along with the tree very well. However, when we look at the checkerboard, we can see that it seems to be correlate pretty well with the sequence, with the species complexity. And so this is the first hint, and we did careful examination. So we checked the checkerboard scores against the cell type number. And this is a well-known indicator that the species complexity. We see a very significant correlation, and also it's a p-value significant. And so we have the two measurements, and then we're going to check, okay, so how about the correlation between these two, the global folding and the checkerboard, and then, or you can say that it's a compartment strength. And we can see that those two looks like they are compelling to each other and antagonized. And you can imagine that those two kinds of folding logic, and once you adapt one folding, you cannot fold into the other one. So this is the reason that they have a negative correlation, very strong negative correlation. Because of the evolutionary tree, we can trace the evolution of such the folding structure. For example, here, we notice that for the checkerboard score, and we can say this is the compartment strength, that is continually improving or enhancing, and when the species become more complex. And from the original, that's 0 compartment to 1 compartment and to 2 compartments. And you can see also from the high C plot. And for the global folding, it's interesting that, and first has a random, looks like random in early species, then has a very strong global folding. But once species get very complicated, and then they start to have the so-called compartment or checkerboard scan again. So this is what we deduce the evolutionary history of the global folding. Okay, so when we compare these two, the animal kingdom and plant kingdom, we notice that, so for these two kingdoms, once a new phenotype or new pathways has to be formed, so the animal kingdom has to do something very different. It chooses to have a spatially close in the gene. So the same gene in the same pathway, they will tend to spatially close in the genome. However, for the plant genome, it's a different story. So you tend to make copies and have extra copy and make lots of gene clusters. And you can see very significant, and they're using different strategies. And since because plants has a very strong global folding, and it's very easy for plants to have a whole genome duplication, to have a hybridization between two different species. So in compared to the animal kingdom, it's pretty rare that it's very hard for them, because once you have the whole genome duplication or hybridization, it will disrupt these spatial regulation patterns. And okay, so this is the final slide that. And so we also look into the development of the cells. And so we take the human embryogenesis data, and it's from the other people. And we notice that when it's in the 2-cell stage, it has a very strong global folding score. And once you reach the 8 cells, and suddenly the global folding is completely gone. And then when the number of cells goes on, when species the cells become differentiated until 6 weeks. We can see that horizontal, vertical, the checkerboard pattern become very much dominant. And all right, so this is the summary of the slides. That's, today I'll talk about a sequence to image, and we can use the one-dimensional sequence convert to two-dimensional image. Then we can enable effective coding of data and knowledge. And second thing is that we can demonstrate that we can use all kinds of image operators to do local, distal, and chromosome scale classification. And also talk about one discovery that we can use the sequence to image approach to analyze 3D genome evolution. And finally, I would like to thank Darwin Tree of Life for providing such a wonderful data set, and also thank the funding agency and also the 4 really excellent PhD students. All right, thank you.
We have time for a couple of questions.
Yes, very good talk. I have like 2 questions. First is the evil. You said you borrowed the idea from Hi-C. So my question is, so how do you define the interaction if you do not use Hi-C data?
So we have to do some measurement, right? So the interaction we have to gather because it's a 3D-dimensional genome.
Oh,
Yeah. Okay. You mean annotation?
Yeah, in an annotation. Yeah.
Okay. Yeah. So the idea. So we actually borrowed the idea from Hi-C, but we don't need Hi-C data. We just learn from the frequency, whether some DNA segment or DNA bins that actually show up together when we scan, because it's a labeled data set.
Oh, so basically, this pixels represents the represents what?
Yeah, one bin is 32 base pair, and if there's a co-occurrence in certain genes, and you will have a higher probability.
I see. So
Yeah, and because we have lots of such instances, we can learn the frequency.
So is that the bin size matters or not?
Bin size doesn't matter very much. This is just one parameter.
Okay, thank you.
Hello, thanks Kaya for the very nice talk. I'm Jin Miao here. Thanks for the nice talk. So I like the idea of sequence to image conversion. So have you applied your approach to the cancer genome and to see how the cancer's genome has evolved from the onset, progressions, and recurrence? Thank you.
Yeah, thank you. So you can look at it, the somatic one exactly target for the cancer genome. And indeed, we are collecting a lot of data sets using long reads. And at the same time, we also try to apply this technology to short reads. And because there are plenty of short reads in the field, in many hospitals, in researchers' lab.
Yep. So have you looked into the single cell DNA seq of the tumor? Because tumor cells are known to be very, very heterogeneous, so they may have different mutations in video cells.
Yeah, yes, indeed, we are working on that. Yeah, we have some preliminary data showing that we can able to look into the cell level mutations and correlate with the expression values.
Yeah, thank you so much, and looking forward to the new discoveries.
Hi, Dr. Ye, exciting talk. I have follow the questions. So based on your this sequence image or something, can you predict this cancer genome? How do you say the CNA copy number variations? Those, or even follow the circular DNA? I think you may know it.
Yeah, so those concepts are basically from the short reads, and so when we apply long reads, and we can really decept whether the CNV, and because CNV we don't know the precise breakpoints. Using long reads, we can precisely pinpoint the start end of the copy number.
How long it is?
And depends on whether you are applying the raw long reads and 20 kb, maybe 1 million.
You mean the
if it's a assembled context, we can detect even longer mutations.
Okay, so second generation sequence you cannot predict that.
And we are working on that. So this is an active collaboration project with Illumina.
I see. Okay, yeah, looking forward to your new.
Okay, very exciting talk. I have an open question. DeepSeek released the DeepSeek OCR several days ago. The core of its algorithm is to convert text into images, which is somewhat similar to your work, right? The algorithm greatly increases the number of input tokens and improves algorithmic efficiency. What's your opinion on future direction of compressing sequences into images?
Okay, thank you. This is actually exciting that I just learned that DeepSeek also able to use ORC to compress the input. But in our point of view, that biological sequences, for example, DNA, only has 4 letters. And in a way, it has to compress a lot of information. For example, the epigenome and also the long-distance coding for the gene coding for the regulatory, and also for the 3-dimensional structure. So in the way that DNA sequence has a multiple dimension data compressed into a string that's with so little alphabet. I think it's a very different way to think whether we should, because English language has many words, and also each word has different letters. But DNA sequence is only had 4 letters. I think we have to invent something different to really able to compress, can also decode all kinds of disentangled information. Yeah, and I think as a sequence image is just our way of doing this, and we can have the knowledge, existing knowledge, and coding together with the data. I think this is all of philosophy. So because all the previous information, all the knowledge is already in the field, we cannot just using data-driven, just straightforward to take DNA as throw it into the language model and pretend we don't know anything. I think when we have the sequence of the image, we can cleverly code the sequence together with the existing knowledge embedded. For example, we can have the methylation, we have to have the 3D genome information all coded in the same space. And we can use straightforward CNN to have all kinds of calculation. Yeah.
Okay, thank you, Dr. Ye. This was a very nice talk. Okay, so with that, we come to an end to our morning session. Let's thank all of our speakers. And we can start our lunch now, and we'll resume at 1 o'clock. So enjoy lunch.